Hello, my name is Prabha Ravichandran. I'm a research biologist working for Agriculture and Agri-Food Canada in Lethbridge, Alberta, Canada. The topic of today's discussion is estimation of leaf nitrogen content with leaf spectrometer in potatoes. Nitrogen fertilizer is one of the essential plant nutrients optimized to enhance crop yield and quality in modern agricultural fields. The conventional practice of applying nitrogen in a split dose before planting or in a random split dose without having precise knowledge of the amount and timing of the crop nitrogen requirement has led to excessive use of nitrogen fertilizer in Canadian agriculture. Excessive nitrogen application is a serious environmental issue and has a huge impact on greenhouse gas emissions, soil health, and water resources. So the objective of this study is to see if we can estimate the leaf nitrogen content in potatoes, which is at a different rate of nitrogen fertilizer, and whether we could use a leaf spectrometer called SpectroView to estimate the leaf nitrogen content. So let's look at the experimental design. So we used a commercial potato cultivar called Russet Burbank for this study, and this experiment was conducted at Agriculture and Agri-Food Canada field in Frederick, New Brunswick. So the treatment includes an untreated control along with nitrogen applied at 5 different rates, 47.5. So these numbers were chosen based on the soil analysis we performed before the study: 47.5, 95, 142.5, 190, and 237.5 kilograms per hectare. Leaf spectral profile were obtained using the SpectroView leaf spectrometer, and it has a range between 360 to 1100 nanometers at a range somewhere between at an regular interval of 0.5 to 0.7 nanometers. So here's an aerial view of our field. So here we have 4 rows of the actual treatment and some guard rows in between. And here's the methodology. So we took the leaf spectra at 3 different stages: at stolon initiation, tuber initiation, and maturity stage. And we picked the 4th leaf of the plant in rows 2 and 3. So we let go of the edge rows of a for-loop plot. And for analysis, after taking the spectral profile, we picked 24 leaves per plot and dried it at 60 degrees Celsius until the weight of the sample is stable for 24 hours. And 0.2 grams of the dried sample was then passed through a 0.2 millimeter sieve and then used for analysis. The total nitrogen content of the sample were analyzed using dry compression method in Licotromac CNS analyzer. The calibrated spectral profile from the SpectroView leaf spectrometer and the ground truth from the analysis were used to develop the partial least square model. And let's look at some results. So here are the here is the visualization of the 72 spectra of the leaves that were sampled at 3 different stages. And the data set was randomly split in the ratio of 0.8 to 0.2. 80% of the data set was kept for training and 20% was kept for test. And the model was trained using the training data set and optimized. We optimized the number of components somewhere between 1 to 30. That was the range that we used, and the numbers you see here is an average of 1,000 runs. So here is a small graph representing the trend of training and test coefficient of determination, R square, with changing number of components. So if you see as few as 5 components were sufficient to get an accuracy of 78.6%, and pretty much after that we did not see any difference in the performance of the model. So that is our result. So we achieved a maximum R square of 78.6%. And we just tried, so this is a preliminary study, we just used partial least square regression, and we are having a few other models like artificial neural network and deep learning models to further explore. But like since we had like very less number of observations from the first year of 3-year study, we were limited on the number of models we could use. We are hoping to collect more data in year 2 and 3 and kind of combine them together and try different models, and we will be more than happy to share our results from from year 2 and 3 in the upcoming conferences, CRISPR conference. Thank you very much. If you have any questions, please let us know. We'll be happy to answer them.